A very good morning to you, Willem. How are you?
Good morning, Fredos. Apologies, but in my calendar, the timing was slightly different, but I'm there.
It's all right. It's fine. I'm glad that we could connect.
Sure.
Yeah.
I see there is a very large class of very many people.
Oh yeah, there's a group of friends from the Fakara Health Institute, my colleagues, and then we have some of our friends also from Cambridge. You know Eric Ochomo's group?
Yes, yeah.
Yes. So I think many of them are just like me, very excited to be able to have this conversation with you.
Okay.
And,
I had not understood that you would invite so many people.
It's all right. Don't worry about it.
Okay.
Yeah. So just for your information, my colleagues are staying on the call. They are all muted, but if anybody has a question, they'll post it on the chat, and
Sure.
we'll let you know. But ideally, we're just going to have a conversation. We have a number of questions, and we hope that we can take this in the most relaxed way. It's something I wanted to do, like I told you last time, for a long time. So it's exciting to be able to do this. But first, just thank you so much for making time, and apologies for the slight miscommunication on the calendar.
No problem. I would say just go ahead, and I'll listen in, and if my interaction is required, please don't hesitate.
Mostly it is.
But so I should keep my my speaker
you should have your video
on unmute. Yeah, my video is on.
And your speaker as well, just the way it is now.
Yeah.
So my speaker and your speaker will be my video and your video will be on. Everyone else will stay on mute. So
and you can do the screen sharing.
And I can do this screen sharing. Yes.
Yep.
I'll do that.
Fine.
I'll do that. Yes. So just one quick moment. I'm gonna do. Yeah. So, Willem, again, like I said, thank you so much. And part of the conversation that I would like to have with you today is really the historical perspective of the vector entomology, malaria entomology. And the last time we spoke about this, we had discussions about how it all started and your days in Ifakara many years ago. And the time we started to work together and until now. But to begin with, Willem, I want us to begin in 1980s, in the early days, your early days of malaria entomology in Africa. And our first question really is just for you to reflect a little bit on how the situation was at that time for medical entomology in Africa.
Okay, thank you very much, Fredros, for this question. In the first place, welcome everyone to this interactive seminar this afternoon. And I hope that if you have any question, please put them in the chat room so that Fredros or I can later answer them. But coming back to Fredros' original question, my career actually goes back to the very early days of my career as an entomologist in Africa. Goes actually back to the early days when I started working in Mozambique for the United Nations. That was in 1980. And as many of you know, Mozambique had actually become independent in 1975. But with the independence, all the knowledge that was in the country actually disappeared because many of the knowledgeable people actually left Mozambique for South Africa or Europe. The country at that time literally had only one person with an academic degree. That was 1980. And I was just a fresh junior medical entomologist, fresh from university, and was basically asked to revamp the trypanosomiasis control organization of Mozambique, because both human and animal sleeping sickness, or animal trypanosomiasis nagana, were considered a major obstacle for rural development. So off we went, and for 3 years I crisscrossed the entire country, and at that time we were very fortunate that we could at least train a whole series of technical officers who knew how to recognize tsetse flies and how to collect them. And we had no molecular tools at all to find out whether these flies were infected or animals were infected. You basically had to take blood or squeeze the fly and see whether you could identify the trypanosome under the microscope. From Mozambique, I moved on to Nigeria, which is here in West Africa, to continue again with a program on trypanosomiasis control. And from there, in 1987, in 1986 actually, I was invited by the United States Department of Agriculture to come for a academic year and actually refresh my own knowledge on medical entomology. And it was there that I became very interested in mosquitoes, because mosquitoes are, of course, not only causing a problem in Africa, but they are a huge problem in South and North America. Yeah. And the Americans were already by then deadly afraid that mosquito disease might actually invade the country, as it has done since that time. But I became very interested in mosquitoes and was actually curious why the only method of collecting a mosquito was with a light trap and solid carbon dioxide. And that alerted me when I got back to Europe, and via Europe back into Africa in 1987 in Tanzania, that I thought what we basically need to know if we actually want to get a better grip on the transmission of malaria in the African continent, we must have very solid methods of mosquito collection.
So your first interaction with malaria work happens in Tanzania in the late 80s?
That happened, well, not the first interaction, because in the 7 years that we lived in Africa, I contracted malaria 5 times.
Wow, sorry.
And it was, each time it was falciparum malaria, but fortunately there was no drug resistance at all.
That's good.
So chloroquine usually actually treated you quite well, because the chloroquine resistance actually came in only in the 1990s. But nevertheless, I can tell you, like most of you actually have experienced yourself, it's never a nice experience. And this was in spite of taking prophylaxis all the time. It was not that we were not taking preventive measures. We slept in a bed net. We took prophylaxis, and in spite of that, we had a rather high risk. And one of the risks was, of course, that as an entomologist, you are often out in the dark, outside, because that's when you need to catch your target organisms.
Yeah. So is it this malaria situation that you had? Could we trace in any way the fact that you had this malaria infection so many times, that you are also so strongly interested in medical entomology from on malaria side? From
well, that interest became stronger and stronger, but it already actually was initiated in my student days back at the university.
Yes.
Because I spent a year in Zambia as a training period.
Mhm.
And I was just a general biologist or entomologist at that time. But I happened to tag along with a wildlife veterinarian who collected insects that were very important for wildlife.
Right.
Not only mosquitoes, but also ticks and biting flies.
Mhm.
And when I understood that also these arthropods are responsible for transmission of all kinds of animal diseases, my interest in medical entomology became really strong, so that I actually decided to do a PhD in it. And I think I've never lost it since that time.
That's nice. Well, today we still have to deal with this situation, and it's very interesting that for diseases such as malaria, we are having to deal with exactly the same problems that people dealt with 30 years ago, 40 years ago. There is, however, one place in history, this is Brazil, where they seem to have been pretty successful in one particular case, going the mosquito way, eradicating this. Uh, I want to ask you, as someone who I know is also very focused on integrated pest management, and we will talk about that at a certain point. What do you think are the key lessons that we can learn from some of the initiatives that were taken by Fred Soper and crew in Brazil? What can we learn from that would be relevant for the current African situation?
Well, firstly, we should all realize that before 1930, there was malaria in Brazil.
Right.
But it was mostly in the coastal areas and transmitted by a mosquito called Anopheles acausalis. And it was more Plasmodium vivax than Plasmodium falciparum. And it was found all over South America, actually, in the coastal regions.
And this is Anopheles aquasalis.
Yeah.
All right. Yeah.
That was the species that was responsible for the transmission. Even cities like Rio de Janeiro and Sao Paulo had sometimes huge outbreaks and many people sick. Mm-hmm. But as you well know, vivax is less fatal or less invasive than falciparum. So most people actually recovered from it, even though there was not a very good drug. But then, by accident, and we still don't really know how it arrived there, in the early 30s, another type of malaria all of a sudden was spotted in the northeast of Brazil, which is a very dry, almost desert-like area. But along the waterways, all of a sudden people contracted malaria, and that was falciparum malaria. And soon after that, it was discovered that it was a member of the Anopheles gambiae complex. Right. Although no one had any clue that Anopheles gambiae was a complex, because that was only discovered 30 or 40 years later. The mosquito must have arrived with a shipload from West Africa, and it arrived in virgin territory with almost no natural enemies for it. So it could spread very, very rapidly, aided by man-made breeding sites to a very large extent. The rice fields, people kept water containers everywhere. But then a clever man, Fred Soper, had an idea that he could actually eradicate the mosquito from Brazil, because it still had a relatively limited distribution, and outside the distribution area, the climate was so harsh that it was almost unimaginable that these mosquitoes could survive there. And they had only one insecticide, one option available, and it was Paris green. And as we now know, that's a highly toxic substance, certainly for all kinds of aquatic organisms and humans.
Okay.
Nevertheless, if as a powder you sprayed it over the water, the mosquitoes were dead within 24 hours. And he got a military-like operation organized, just like they did along the Panama Canal 20 years before. And lo and behold, by 1940, the mosquito had disappeared from Brazil.
Okay.
Now, what lessons can you learn from the Brazilian operation if you translate that to Africa? Because that's your question, isn't it?
Right.
What we see in the African environment is that over the evolutionary times, members of the Anofelis gambiae complex have evolved to such an extent that they actually have an exquisite ecological relationship with their environment. If you look at members of the Anofelis arabiensis, they can survive in the dry desert areas of Sudan, in the dry valleys of Ethiopia, but just as well in the very hot, wet valleys of the Kilombero Valley in Tanzania.
Right.
Suggesting that this is a highly adaptable species, just like Anofelis gambiae sensu stricto, which we found also all over tropical Africa after a while. And the difference with the South American climate, of course, is that, as I said, the climate was actually rather unsuitable for this mosquito to spread or to disperse into very wider areas.
Right.
So the contrast is, if you look at the gambiae or mosquito gambiae complex situation in East Africa, for example, the only places, to my knowledge, where you do not really find it is in the highlands. But we know it can be found up to 1,500 meters quite readily. Right. Like if you look at Usambaras in Tanzania, or even in the highlands of Kenya, around Kericho, you easily find it there as well. Yeah. And where you don't find it in East Africa is in Namibia, in a desert situation, because there's simply not enough water. So here we're dealing with a species that over the centuries has adapted itself so very well that it is an extremely difficult task to say, can we achieve a similar eradication as SOPR did in Brazil? And my personal answer would be no, I don't think so.
And I think that's an important point actually to bring out of it. So it is now, I've read recently, and I don't know if this is a consensus, but it is now believed that the mosquito that invaded Brazil was actually Anofelis arabiensis.
Yes, yeah.
And that the situation would have been different if this had been Anofelis gambiae sensu stricto. The other difference is that in Africa, unlike in Brazil, you have more than one vector species that is very competent. And this brings me to the question of Anofelis funestus, which is also a big deal, especially in the Eastern and Southern Africa region, but somehow is not so much discussed. And one of the things I'm gonna request you to talk about is anthropophily. But if you like, we could start from the point of why is this vector, which is otherwise quite competent, so neglected by biologists?
Thank you. Why is Anofelis funestus compared to Anofelis gambiae so neglected by biologists? Right. There are a couple of reasons for that, but we should realize that the study of the biology of African anophelines really took off only after the Second World War, in around the early 1950s, right. With Gillies working in in Tanzania and French groups in West Africa. And at that time, they basically started collecting the species. And since the densities of Gambi, particularly Gambi sensu stricto, were so much higher than of Funestis in most cases, they actually naturally focused their attention on it. Although Gillies, to give him his dues, also has written a lot about Anopheles funestis. He very well recognized that the two were very different species, had also slightly different behavior and ecology. But Funestis had the enormous disadvantage that nobody managed to put it into culture. Whereas the minute you took Gambi or Arabiensis, you were usually highly successful and could raise them quite easily in the laboratory. So you could also study their biology, the number of eggs they would develop, how often they would mate, how long they would live under certain circumstances, and so on and so forth. And I think that's probably the main reason why Funestis was neglected, whereas we now know that Funestis is probably just as important, if not even more important, a vector than Gambi.
Right. I mean, we spoke to Derek Childwood, and he was saying, "Hey, you got to read Gillies' work."
Yes.
On that,
yeah.
Like it seems like people kind of not only neglected the vector, but also neglected the early writings.
Yeah.
About
yeah, I can only concur with him because I believe it's required reading actually to go back to Gillies' work from the 1950s, what he did out of Moesa. And then lots of people would say, "Oh yes, I recognize that. I wish I had known that earlier."
Yeah,
because it deals with microlease studies, it deals with house entering studies, it deals with outdoor biting, daily rhythms, anything.
Actually,
you
you publish something and then you realize Gillies already wrote it.
Yes, exactly.
Yeah. Thank you, Willem. In addition to Anopheles funestus now, very unfortunately, Africa is probably going to be dealing with this other guy.
Yeah.
And you and Professor Lindsay, you have been very vocal about Anopheles stephensii, and you've written about this. You've warned scientists about this. I was going to ask you whether we should worry, but I know you will say yes to that. So, I mean, what should we do about it? Maybe I should ask that.
maybe you should ask, why should we worry?
Why should we worry about it?
Well, in the first place, I'm sure that many of the audience this afternoon, who all are from Tanzania and Kenya, probably do not know the epidemiology of malaria in South Asia. But in countries like Pakistan and India, Anopheles stephensii is probably the most important vector, next to one other outdoor biting species. But the reason that stephensii is so important, it's an urban vector. You find it in big cities like New Delhi, Calicut, Rawalpindi, Lahore, and so on, these big urban centers. And why do you find it there? That is because the authorities fail to provide everyone with sufficient drinking water. So for security, most people build in their house basically a water tank on the roof, and that's inviting this mosquito to come around your house.
So Anopheles stephensii breeds exactly in the same habitats as Aedes aegypti.
As Aedes aegypti, right. But it goes even a bit further. If you have, for example, a vegetable garden, you have a big water tank there, like in the middle of Dar es Salaam, and people keep water there to water their plants with, for example. You can find huge populations of stephensii there, should stephensii become established in Tanzania. So the reason that Steve and I wrote this article and actually alerted the World Health Organization about it, that in our view, if we don't do anything about the arrival of the species in Africa, we're going to see an incredible surge of malaria in most of the big urban centers like Mombasa, Dar es Salaam, Maputo, but possibly even Nairobi, and Kampala, you name it. Every big urban center is at risk.
Yeah, we have greater connectivity now with the rail lines, connecting the inland ports into Uganda and Rwanda as well. So of course that's probably too much extrapolation, but I get the point.
And there is a fairly nice article from the Liverpool group and the Oxford groups. With, I think Sinka is the first.
Marian Sinka, yes. This is a PNS
where they actually predict how far Stevensite could spread into Africa if it was uncontrolled. And I think we should really worry because this species could have been controlled quite easily because you can identify the breeding sites. It's far less often found in rain puddles and so on. But it so happens to be in areas that are in conflict zones. Yeah, think about northern Ethiopia, of Sudan, Somalia, all areas where actually people rather would not go.
So how do you think we should be monitoring in terms of if we wanted to know that Anopheles stephensi has arrived, if we wanted to mount control measures in advance, what should we be doing as entomologists in East Africa about this?
Well, you know that one of the pillars of the global vector control response is increased surveillance.
Right.
And I would personally, I would start immediately with that, if there are enough people to do it.
Right.
And basically teach people what the species looks like, where it breeds, where it's expected to be, and put out scouts in and around Mombasa, in and around Nairobi, and all over northern Tanzania and eastern Tanzania, where you have sort of relatively sizable urban centers like Tanga, Dar es Salaam, and so on.
Right.
And start with that at least.
Yeah.
It doesn't have to be very costly. You at the same time also get the information about where would you find Aedes aegypti. And there is of course a lot of interest in Aedes aegypti at the moment because of its ongoing expansion of dengue and possibly even chikungunya. So you get two kinds of information that you want to know.
Just one last question about that. And actually this is related to our second point, which is about your book, the parasite vector interactions. Before we answer this though, there's a related question which touches on Anopheles stephensi. Tell me about Anopheles stephensi and P. vivax versus Anopheles stephensi and P. falciparum. How are these interactions, and are they equally as bad?
Well, stephensi turns out to be an excellent vector of falciparum.
Right.
But it's also a vector of vivax. We know that, and in many cases you can find double infections in mosquito. Because of the difficulty of culturing vivax in the lab, it needs to be, it prefers actually a lower temperature than falciparum. Right. Falciparum is usually held at 25, 26 degrees Celsius, whereas vivax has an optimum of between 18 and 20 degrees Celsius. It's not so easy to study these parasites live, so to speak. Nowadays, a lot of done is via molecular tools, but you still would like to know what is actually the possibility that a mosquito, once infected with one of these parasites, can actually also pass it on to somebody else. So what is its infectivity? Concerning your, so that's the situation. That's the situation you see in Asia. I think in Africa, perhaps with the exception of Northeast Africa, like Eritrea and parts of Ethiopia, where vivax is found, you do not find vivax very often in Tanzania and Kenya, largely because of there's a genetic reason, of course, the Duffy coat of the Duffy characteristic that many of you carry, which is rather unsuitable for vivax development. So if Steve and I would come to East Africa, my concern would really be falciparum.
The falciparum one. Okay.
And falciparum in urban centers where many people may never have been exposed to malaria parasite.
I see that. I see that. That will, I see that. So in that case, it's most likely going to appear as outbreaks, even in places such as Dar es Salaam that are supposed to be endemic.
Yeah.
Okay.
Now, concerning your question, who was there first?
Who was there first?
The parasites or the vector? Of course, with
there are some evolutionary ecologists with us on the call today, so this is an interesting question.
Probably the parasite was there first, we think, because malaria, of course, emerged as an avian parasite. And then went on to also move into people, where it became a rather specialist parasite. But we know that mosquitoes have been there for millions of years.
Right.
And I think that we basically still need to wait until we find maybe something in amber, where you can identify the parasite from an early mosquito. I'm not sure whether it has happened already, but we can safely say that already more than 50,000 years ago, there was a well-established relationship between Plasmodium and Anopheles.
All right, okay. I've had recently a very interesting argument about the egg and the chicken. Following exactly the same line, that a lot more animals can lay the egg than just chicken. So the reptiles can lay eggs too. And that means the egg came first. And I guess it's the same thing here.
In the case of Plasmodium, you have to realize there are hundreds of avian Plasmodium species.
Right.
Whereas at the moment, we actually only know of 5 Plasmodium species that have adapted to man.
Right.
To humans.
I spoke to Pierre Canaval, and I'm hoping that I will speak to him again. And, being European, he was very keen on explaining that actually Ronald Ross worked on bad malaria.
Yeah.
And it was almost dismissing that, Ronald Ross was never an entomologist. In fact, he never worked on human malaria that much.
Now, Ronald Ross was a medical doctor, actually a failed doctor, and he was sent off to India by his British boss, who told him, well, if you don't know anything to do, I would like to see if you go just and find out where the mosquitoes play a role in the transmission of malaria.
Right.
Because, and this was Patrick Manson who told him so, and Manson had discovered 20 years earlier that Culex species were responsible for the transmission of filariasis. And Mansonia mosquitoes, actually, and he did that in China. So Manson all along suspected that mosquitoes were actually involved in the transmission of malaria.
Right.
And Ross actually had no idea what kind of mosquito he collected. But if you read his papers well, this was avian malaria and Culex, with which he won the Nobel Prize.
Right. Yeah. Pio was saying that he actually, in his writings, he called them brown mosquitoes, and he named them by color. He didn't name them.
Well, what you actually have to read is work from two Italian scientists who worked at the same time as Ross, also in 1897, 1898. And they discovered that anophelines in Italy were the true vectors of Plasmodium falciparum. It's never understood why they did not share the Nobel Prize with Ross, but Ross was apparently the first one who got his article accepted in Nature, and that got him the Nobel Prize. But in fact, it should have gone to these Italians.
To the Italian doctors. Thank you so much, Willem. I want us to reflect a little bit on a place that you lived before Ifakara. So, of course, this is data that my colleagues, Lina and colleagues, have data they've collected from Ifakara recently. They did 3,500 trap nights and found only a single infected Anopheles. It was an Anopheles phonestus caught at 3 a.m. One night outdoors. And it was the only one that was infected with the parasite, returning very, very subunit EIRs. They then went to the same villages nearby, and as you can see, confirmed that there's nearly zero malaria in the middle of the town and in the neighboring areas, even though you still have prevalence rates of up to 43%. Now, these are areas that not so long ago were considered a very, very highly intense transmission, very highly endemic. When you see a picture like this, does it give us any hope in terms of whether malaria can actually be eliminated gradually in some of these locations?
Oh, it definitely does, Fredros.
Right.
I think two things are at stake here. When we started work in Ifakara seriously on malaria vector ecology and malaria entomology, it was around 1988. At that time, the Ifakara Institute was really very small, only 2 buildings, and sort of an outer barracks that we used as a laboratory. But we started working in the Namawale area for a couple of years, and I did it with Derek Childwood.
Right.
Edith Limo joined the team, and she was our first PhD student, and she basically studied egg-laying behavior of anophelines there. But what was the point is when we started collecting mosquitoes around Namawala, the house structure of the villages was very different from what you see today.
Right.
I would say not even 5% of the houses had a solid roof. Most houses were really still built of sticks and mud, and would survive 2 rainy seasons, and then you would have to build a new one. So mosquitoes had at all times actually free access to these houses.
Right.
And what is also very important, farmers were so poor that they actually had no access or money for agricultural pesticides.
Right.
So they grew their rice or cotton or maize or whatever, and they basically had to suffer the consequences if there was an insect plague or a disease. In some houses in Namawala, we collected up to 2,000 blood-fed mosquitoes every day at the height of the rainy season. Imagine that.
Oh.
2,000. And Derek and I published that. in the early 90s. So it was no wonder that when medical staff started analyzing blood samples of the villagers, that we found that throughout the year, 80% of the population carried malaria parasites.
Right.
And because of the huge number of mosquitoes that were there, the average infection rate was still not much higher than 3 to 3.5%, because there's a very high mortality rate early on, and only few mosquitoes went on to reach an age where they could actually transmit more than 15 days. If you compare that now with 2000, then we're 20 years on, approximately. Right. Tanzania developed quite rapidly. House structure changed actually dramatically. You know yourself, if you now return to the Kilimanjaro Valley, that most houses have a proper roof. Many houses now also have some kind of a window. Grass roofs are completely, well, I would say not eliminated, but rare, if they're still there. Most houses also have a door. So it's more difficult for mosquitoes to get in. The insecticide-treated bed nets became available in the Kilimanjaro Valley as of the mid-1990s, approximately. First, not very many, but by around 1997, 1998, there was a fair distribution of them, and when people discovered how well they protected you, most people wanted them, actually, also because they were no longer bitten so much. And secondly, I think, and this is difficult to prove, but pyrethroids also became in as agricultural pesticides. Yeah. At the price where they were quite sort of more affordable to farmers than ever before. And we must accept that if most of the mosquitoes actually in the rainy season originated from the rice fields, then they didn't get much of a chance if these farmers used these pesticides. Even though they sprayed maybe 4 or 5 times in the spraying season, it nevertheless every time meant probably a knockdown of these mosquito populations, what we don't know, 50, 60% or so. But nevertheless enough to have a dramatic decrease in mosquito densities. Yeah. And I think these two, or actually 3 activities together, house improvement, the availability of treated bed nets, and modern farming technologies may have sort of conspired to get this actually rather nice effect. Right, yeah. No, thanks. You talked a lot about mosquitoes entering houses in search of blood. Mm-hmm. This is also a subject that you have written very widely about. And I pulled up this figure from your 2013 paper with Neil. Yes. Where you compare the preferences of different mosquito species to bite humans. And Anopheles funestus and Anopheles gambia says way up there, mostly Anopheles funestus being really atrophilic. We also pulled out a graph that a colleague of mine called Alex and myself worked on at the request of Dr. Jerry Klein to map the human blood indices for mosquitoes around the world, which shows very clearly that the mosquitoes you find in Africa have pretty high human blood indices. Why is this important? Why are these host preferences and host choices important for mosquito biologists? A related question there, which you can answer if you like, is what would be a perfect experiment for us to study this in the field? Okay. Well, let's first answer, let me try to answer the first question first. Why is it actually that only very few mosquito species worldwide have evolved to have such a very strong association with humans, with Homo sapiens?
Right.
And I mean, the study you show here, this review, demonstrates it rather convincingly, particularly for Gambi funestus and Aedes aegypti. Now, we believe that over evolutionary history, these mosquito species underwent an evolutionary change by establishing themselves with human beings.
Right.
Originally, human beings did not live in dwellings, but by the time they started developing houses, these houses created actually an excellent resting site and a safe resting site for these mosquitoes, because it was dark, it was humid, they only need to fly 1 meter to feed themselves, and they've profited from that enormously, and could actually produce optimally at the end by feeding on human blood. Because they had the only actual natural enemy they had in these houses were the human beings. But since it was so dark, most people actually hardly noticed that, because you do not notice the bite of an Anopheles gambi or an Anopheles arbiensis.
Oh, really?
No, compared to Aedes aegypti, just try it once and you realize it immediately.
Yeah.
I can continue sleeping in a house with Anophelines, they don't bother me. Whereas as soon as there is only one Aedes aegypti in the room, you notice it.
You notice it, right? Yeah.
So in the course of evolutionary history, these anthropophilic mosquito species, as we call them now, have actually lost the ability to survive in an environment where people are not present. Mm-hmm. Just go into any wildlife reserve in Kenya, Tanzania, or Uganda, and you will definitely not find Gambi sensu stricto or phonistis, because they have no way of feeding. But with Arabiensis, it's completely different. Arabiensis is an opportunistic species. Arabiensis will feed where the population density of humans or an animal is highest. Right. Look around farms. If farmers have a lot of cattle, many of the Arabiensis will actually feed on the cattle. However, if there are few animals in a village, they will rapidly switch and feed on humans. So that's a major difference between Gambi phonestis on the one hand and Arabiensis on the other hand. If you go to Aedes aegypti, which is there in bottom left, that species is just like Gambi. It can actually not survive on wildlife or animals. It needs humans.
Right.
On the one hand, that's a risk for these species, that they become so specialistic that it's also easier to get rid of them. The Arabiensis can simply go somewhere else and establish a new population. And Gambi cannot do that. And it is my conviction that one of the reasons why the Gambi populations have collapsed so much in East Africa is because we've been so successful with these bed nets, and we don't give them any other opportunity.
Right.
Whereas Arabiensis populations are still relatively, or they can be relatively high. I know that personally from the Ahero region in Kenya, where the densities are still as high now as they were 25 years ago.
Right.
Then comes, of course, the intriguing question, and what about phonistis? Why do we still see phonistis?
Why did it not disappear with the
exactly.
MBSS?
And this I think it is the prime question that I would challenge all of you. Start studying phonistis and the biology of phonistis, and try and find out why can phonistis actually be so extremely successful in spite of all the attacks that we are putting on it.
Right.
House improvement, so that it's more difficult for them to get to their hosts. Bed nets, even more difficult for them to get to their hosts. I have no answer to it, honestly.
Well, we have some of my colleagues have examined this. We only have hypotheses, and one of them is that Anopheles funestus, being much smaller, developed insecticide resistance much earlier and much faster. So that by the time you had the bed net come in, it was already established there. And unfortunately, in many insecticide resistance surveys, you hardly get Anopheles funestus because the aquatic habitats are not that easy to find. And
right. Yeah.
the WHO protocol still requires you to collect the larvae and keep it. So many resistance surveys just miss it. And this is probably one of the reasons. But we also see from historical records that if you had a good insecticide, especially for spraying, that it will just disappear. So it's an intriguing question, and that's why I really, it's as you challenge us that we should study this much more.
If you look at the Tavete Para scheme from the 1960s, that was very successful for Funestus, just like Funestus control program in Madagascar, where the species almost disappeared. But because of the resistance, it also came back with a vengeance.
Right.
And it's extremely intriguing why it's apparently far more resilient than Gambisensis strictum.
It is.
And that may already actually also give a hint that the evolutionary history of Funestus may well be different from that from Gambisensis strictum.
Yeah, there was a paper from Nora Byzanski's lab yesterday or maybe 2 days ago that suggests they have a lot of introgression from the sibling species in the Funestus complex. I have to read this again. But,
yeah, please do.
And apparently this helps with its vectorial capacity as well.
Okay.
Anyway, Willem, there is a related question here, which you have also studied quite a bit. The mosquito and their sense of smell. We know now that mosquitoes are extremely reliant on smells, attractants. And, one of, I think, perhaps your most widely read article is the odorant behavior of the Afrotropical malaria.
Yes. Yeah.
So the figure at the bottom left is from that annual review. I think it was in 1999, I believe.
Yeah.
How much do we know about these processes now, and where are the gaps?
Okay, thank you. When we wrote this article in 1999, we had almost no real understanding how the biological process of odor reception went. The only thing we knew was the strong behavioral responses of Gambisensis strictum and arabiensis, and particularly Gambisensis strictum. There is a beautiful paper by McGillis from 1964, which he did in Moesa, where he built an artificial house, or he built a house with 2 rooms. And in one room, he had one person sleep, and in the other room, he kept some calves, young cows. And then at the opening to these 2 rooms in the evening, he would release unfed mosquitoes. And the next morning, he would actually collect them from the rooms and would find out where they had gone. And lo and behold, this became a Nature paper, by the way. It became very clear that of the Gambisensis strictum, more than 95% was recovered in the room where the person had slept and not where the calves were.
Okay.
Then Mario Colucci, a well-known anopheline scientist who was very active in the 1980s and 1990s, Colucci did a field study in Nigeria, where he collected mosquitoes in the middle of a cow herd. he worked with Fulani cattle holders there and placed mosquito traps in a line through that herd, and then he put one man in the middle of the herd.
I remember the story. Yeah.
You remember that story?
Yeah, but I didn't know it was an experiment.
And Arabiensis was found everywhere, but Gambia sensostricti was only found in the vicinity of this this pastor who was actually looking after the animals. And then finally, I myself did an experiment in Wageningen with Helen Pate, who is now the Gates Foundation. And we repeated a little bit what Gillis had done, but probably a bit more neatly. Helen spent nights under a big bed net, and next to her was a calf in a cage, and she slept on a bed. And every evening we released 200 mosquitoes there. And we did that with Arabiensis quadrinulatus,
right,
and with Gambia sensostrictu. And the result was again that most of the Gambia sensostrictu were found with blood from Helen. Most of the quadrinulatus had fed on the calf, and Arabiensis was sort of 50-50. And these three studies together are such convincing evidence that there are really genetic differences between these three species, which are based on their olfactory mechanism. Right. So then in the 1990s, molecular tools became available also from neurobiology, where it was discovered that on the antennae of the mosquito and on the labellum, small olfactory receptors were present that would become activated once they came into contact with specific odor molecules from the host. Mm-hmm. And we tested that with smells from a whole series of animals, including as well as humans. And each time we tested humans, Anopheles gambia sensostrictu, these receptor cells or specific receptor cells gave very strong activities. Then we unraveled that and identified 300 chemical compounds that were emanated by a human being and that a mosquito could potentially recognize. But of these 300, only very few were so specific that if you combined those together, and in the end we made a blend of 5 of these components, that to the mosquito antenna it was as if a human being was around. And that then became actually the start of developing a synthetic odor bait, and you yourself were very much involved in that in Ifakara. I don't know how many fatty acids you've tested in 2 years' time.
By my count, I did. So I think I have 2 records that I'm very proud of. One is that I have done the highest. I have done the most consecutive nights of human landing catches. I think so, and I have also done the most nighttime experiments with smelly substances. I did 105 consecutive nights. So,
and those were often not very pleasant.
These were not. There used to be a joke. There used to be a joke in Ifakara that you smell like Fred Ross, because people didn't realize that it's the stuff that I was carrying. They thought I was the smelly guy, because I always carried these traps. But
anyway, out of these combined studies, and this was actually very nice collaborative work, by, let's say, the early 2000s, we had one odor blend, and that has now become the Ifakara MB5, the Beta MB5 blend, which is a fairly good proxy of human smell to Anopheles gambiae, and also to Aedes aegypti, I must say. And
I want to ask you about the MB5 first, but before that, I want us to go to that because I have some questions for you about how that has transformed into malaria control. But before that, could you just go back a little bit and explain if there is any role that other cues play, like visual cues, tactile cues?
Well, we now know that thermal cues are very important as well.
Right.
And this is done by the work done in Burkina Faso. It was actually quite recently published by Frances Hawkes and her group, that if you combine the synthetic smell with temperature, you attract almost twice as many as mosquitoes as when you have smell alone. And heat alone does not do it, because the distance over which heat acts is only 10 to 15 centimeters, and it drops very quickly to background. So the way it basically works is you need the odors to bring the mosquitoes in the vicinity of your trap or your host. And then the combination of heat and smell together, and to a certain extent also moisture, are three components that then actually are, to the mosquito at least, resemble a human being. Which to me is actually quite normal, but in the early days we could not study heat sources in the field because we had no way of how to provide a trap with the heat source.
I get it. We're gonna move now to this situation here. This is a picture of you from Rusinga Island, an island that I am myself very fond of, because in early 2000s, I believe 2001, 2002, before I went to undergraduate school, I must have visited every single house in this island, doing an ethnobotanical survey for Dr. Aklilu Seyu.
Okay.
So I must have visited all this. But anyway,
that's about 5,000 houses. So.
Yeah, I went. It took me a year to do this. I went to so many houses, several months rather. What would it take, Willem, to transform the systems that you have developed with the attractants into viable mosquito control or surveillance tools? And I ask you this because I know you've demonstrated this with your team in Rusinga.
Okay, thanks. Well, it's in the first place important to realize that the Salomal's study on Rusinga was, of course, a proof of principle study. We wanted to demonstrate once and for all, and this is also what WHO demanded, that with odor-weighted traps, you could make an impact on malaria transmission.
Right.
And the main result of the study was that indeed malaria transmission went down by at least 30% by the time we had to stop the study.
Yeah, actually, I have that data here, actually. Yeah, I think,
Yeah, you can see that there.
Yeah.
And we had a significant reduction in the Anopheles funestus populations. They were far more numerous than Anopheles arabiensis. So it turned out that by continuous removal trapping night after night, usually within 6 to 8 weeks, the funestus population crashed.
Right.
Whereas the arabiensis population remained more or less, was sort of hovering up and down. Since then, we've discovered that the bait we used was not very effective for arabiensis, where it was highly effective for funestus. So the results could have been slightly different, although many more funestus actually contributed to the transmission, also traditionally, than arabiensis on Rusinga.
Correct.
So this was a proof of principle study that shows that you can use removal trapping technologies, which is actually a pull study. You pull the mosquitoes into a device so that they no longer fly on to feed on humans or go and sit on a bed net. And now your next question is, what is now necessary to expand this and make it operational?
Right.
Well, the first place, is what is necessary, we now know that you can make the odor bait a lot more attractive, stronger than it was in our case. We had a trapping efficiency of about 50%, and by adding moisture and heat, you can raise it to 80%, which from the epidemiological model actually makes a huge difference. Because not only do you get an effect much more rapidly, but also more effectively. Second, you can also change the trap model. The Wageningen group has recently actually managed to develop a new trap, the Matego trap, which is twice as effective as the Solomon trap here, or the Desuna trap.
Yeah.
And they're trying to roll it out in several places in East Africa. They have, unfortunately, this work had not been published, and I'm trying to urge them, please publish it so people know about it.
Right. Yeah.
But I've seen the data, and you have seen the data too, because they tested the trap in Ifakara.
Yes.
And everybody was actually surprised how much better it did.
One of my colleagues in Ifakara, Doreen Syria, was particularly interested about the fact that this trial in Rusinga was so effective against Anopheles funestus. And so we have a small Welcome Trust Fellowship there, actually, that Doreen is co-supervised by Francis, by both Francis and Alexander Hiscox.
Okay.
Yes, so hopefully they will carry this forward, and I'm pretty much excited to see.
Yeah, because the beauty of it is that I think this removal trapping system does not require that an entire village is being covered. What we had in Rusinga, and that was difficult to put in the publication, but you know that yourself, a house consists out of a compound. Most house owners have houses with 3 or 4 buildings, in which different wives live or the children live there. But for economic reasons on Rusinga, we could only supply one of these 3 houses, so each compound with only one system.
Right.
And the strength of the study, in my view, is that in spite of that, that only one of 3 or 4 buildings had an odor-baited trap, the mosquitoes were actually removed from the entire compound, suggesting how strong such a system could be. And what we urgently need, basically, is our 2 or 3 other field studies that repeat what we did in Rusinga and get the same result. Because the beauty of it is you actually do not need an insecticide anymore. The mosquitoes are killed as they are in the trap, usually because they dry out, or you can put another biological product in there to make sure they don't live much longer. It can be operated by the inhabitants or the occupants of a house. All they need to do is turn on a switch and turn it off again. And many houses are now getting solar panels. And these systems operate with a little bit of solar energy.
The trapping, that the system that you team deployed in Rusinga, of course the trap sits outside, but it also does this removal trapping. So it doesn't really matter if the mosquitoes were destined to go in or not. And in this particular case, it worked very well for an otherwise indoor biting insect. And in the early days when I started working on traps myself, it was always billed as an intervention targeting outdoor biting. And if you look critically, if I look critically at the data that you have generated, it works very well for mosquitoes that also bite insects. So that's my question here. How big really is the problem of outdoor biting, and how best should we deal with it?
Well, I completely agree with you that if you have an effective removal trap, so to speak, a pull system, it would be just as effective for the outdoor biting population as for the indoor population. It intercepts the mosquitoes as they enter a house, but it also catches mosquitoes that would actually fly around the periphery of the house, provided there are not too many people sitting in that periphery, because then it becomes a competitive factor.
Right.
But usually after 10 o'clock in the evening, there are not so many people outdoors anymore. So if then there still would be outdoor biting, then they would actually be intercepted by the trap. That's often not very well understood, but I think it's an ideal combination and combined effect that you may expect here. Right. Secondly, what is very important of this tool is that it is most unlikely that the mosquitoes will develop any form of behavioral resistance. Because why would they develop a genetic trait that is working against them? They still imagine that they go and find a host. And unless, like Arabiensis, for example, they get frustrated because they can't feed there. If the mosquito flies on, it's gone and out of the equation. So you may even expect a rather immediate effect. And finally, in my view, it also works for a single house, because you can sort of protect all the occupants of that house, provided the flight range of your mosquito is not that great. With bed nets, we've always wondered if they can't reach the host, would they leave the house again and fly to the neighboring house?
Let's talk about you brought up bed nets. Let's talk about bed nets a little bit. Something is happening with this, but just one moment. I just want to confirm that our recording is continuing, and I want us to talk a little bit about the bed net, Willem. You and me have had a lot of arguments about what the future of the net should look like. I am trying to figure out something here. I don't know if...
well, we see a net here with mosquitoes, so...
Oh, brilliant. Yeah. So what are your thoughts about this? What should be the future of mosquito nets? What should it look like?
Well, I come from a school at my university where many of my colleagues as entomologists actually work on alternatives to chemical pesticides for crop protection. And as you know, in Kenya, you have ISIPI, who has done wonderful studies in the last 20 years by developing alternative systems for pest control so that you don't need pesticides. And my dream is still that for malaria control, that should also be possible. Because if you go back to the entire history of malaria, since the discovery of Ronald Ross, and we earlier discussed the control of mosquitoes in Brazil with Paris Green, but then in 1945, DDT became available, and that was the start of a whole range of different synthetic insecticides. Each time, within 3 to 4 years after introduction of a new compound, the mosquitoes developed resistance, and the concentrations needed to be increased to the effect that even nowadays, we sometimes have 3 different insecticides on a net in the hope that you can actually manage resistance. Let's not forget that insecticides are also toxic. They're never very safe. And I still believe that it can't be very healthy to have slept for 10 years in an environment where every day, even however low the concentration was, a certain concentration of pesticides was emanating from the net. My philosophy is, and this is my proposal, why don't we go back to the time when the nets were not impregnated with insecticides? Because nowadays we've got very strong and durable nets. The technology has gone so far that you can now develop a net that lasts 3 to 4 years, whereas when we started in Ivacara in the early 90s, a net would maybe last 3 months before it started falling apart. These are enormous differences. And I think we absolutely need to study what is the efficacy of an untreated bed net in a large field trial compared to a bed net that is impregnated with, let's say, 2 different insecticides. So I see definitely a very strong future for net use, but then net use without chemicals.
You realize you're making a very bold claim.
Yes. And
Willem,
it may be easy for me to say because I live in a country where fortunately we do not have malaria and we do not need to sleep under bed nets.
Right.
And it's very different from many of the countries where 70 to 80% of the population is faced with the malaria risk on a daily basis.
Right.
And you must give them, if possible, maximal protection. But in my view, that protection is not given by continued use of an insecticide-treated net. We have alternatives at the moment that we know, if you combine them cleverly, would actually give as much as protection as a treated net alone would do.
Yeah. Well, you know that I agree with you on this subject, but my opinion doesn't necessarily count.
I'd like to speak out here, and I would challenge all participants this afternoon to think this over
Right.
and make their own conclusions.
Right. Yeah. Do you think this is the same fate for IRS?
This is the same fate for?
What about the future of indoor residual spray?
We won't see IRS anymore in 10 years' time.
You think so? You think IRS will be phased out as a malaria control?
It's far too costly.
Right.
And it still induces the same problem as impregnated nets. Because of the high selective pressure on the mosquitoes, you sort of force them in the direction of a selection of resistant strains. And that goes even faster with IRS than with bed nets. Right. And if I'm not mistaken, I think in the last World Malaria Report that just came out this week, IRS only contributes at the moment to about 5% of all malaria vector control operations. So my question is, why would we do it?
Why would we do it? I mean, well, there are people who argue that if you have the right pesticides or insecticides, that it's actually very effective. But on the other hand, it's a pretty expensive thing to do. Logistically, you know, it's not just going around to spray, but you also have to remove people's buildings. The supply chain for some of these chemicals, organophosphates, the one that is most commonly used now, pyrimidine is sprayed almost at the same volumes as DDT. So the supply chain for that is not easy.
No, and on the other hand, if you look at the environmental protection laws and also the Stockholm Convention, which very much looks into the future of several of these chemicals, right. They all tell us, please stop using them because it's only a temporary solution that is not going to help you forever. And it's better to invest in the development of alternatives than continue with this. India is one of the last countries that still uses IRS to a very large extent because they have the only operating factory of DDT. And they even continue spraying DDT when they know that the mosquitoes are quite resistant.
Right. Well, that's
Yeah.
another bold claim you made today, that IRS will most likely be phased out in not so long from now.
Yeah.
Well, William, there's a piece of good news going on in Africa right now. Housing is improving around the continent. A lot of people are getting better houses. How important is this for disease control?
Well, I think the improvement of houses, and this is of course also linked to an improvement in welfare, and income throughout the country, is probably one of the most important developments that we've seen in 10 years. Because what it will lead to, and you see that already, is that there is a possibility that in future you no longer need to sleep under a bed net. These houses that we see in the top left one, I recognize that's a house probably from the area of Ifakara.
Yeah.
That was basically a big mosquito trap in the time. And then if you look at the house on the right side of it, that's probably the one that replaced it. If the owners actually built it so that the eaves were closed or closed with netting so that mosquitoes could not enter,
Right.
that would actually be the end of fertile mosquitoes, in my view.
Yeah.
And the same is at the bottom two images. Yeah, on the left side is an ideal house, probably somewhere in the coastal region, an ideal mosquito house. On the right side, it becomes rather an inimical house for mosquitoes, at least. They would not feel so happy even to rest there during the day, because the houses have also a tendency to become much drier than a house built of natural materials. So if this trend goes on of house improvement in many, many rural areas, then I'm quite hopeful actually that we would see far less malaria than we've seen in the past.
We had already, according to this publication by Lucy Tosting, I was lucky to work with her on this. We reported doubling from 11% to 22% of people in Africa who now live in houses considered improved houses. And actually, when we were talking about Ifakara, and you mentioned that some of these house improvements have contributed a lot, I am equally optimistic that there will be less malaria as a result of just this livelihood improvements, much more than that. Willem, before we go ahead, there's one question that Eric submitted just earlier about the indoor residual spray, and that was what you think about IRS if done by community members. What about that?
Well, if IRS is done by community members, it would be, of course, a lot cheaper. On the other hand, the risk is also very much higher, which is why to date, IRS is not being done by community members, because you have to make them go through so much training.
All right.
Because you see that here, this guy is wearing a cap, gloves, protective clothes, and so on. Because, as you say, these organophosphates that they spray are very risky compounds and unhealthy compounds. The second risk I then see is that some members of the community might take advantage of using the spraying equipment to spray their fields, because they say, well, a double function. At night we spray the houses, and in the morning we spray our fields. So I personally do not believe, but it doesn't mean that you cannot involve the community in your malaria control. In contrast, if you look at what we did on Rusinga, in fact, it was the population that wanted these systems, and they maintained them for everyone. If a spare part was needed, they would actually pay for that. And I've just completed or participated in a study in Malawi where we actually looked whether house improvement would have any benefit to malaria control in a community where everyone had already a bed net. And house improvement was actually done by the community themselves. So we trained them, they wanted it themselves, and it was a huge success. So there are several options where you can involve, and I think you must involve the community to make it sustainable.
Yeah, that's another subject I want to talk to you about going forward, Willem. We titled your masterclass a transition from pesticides.
Mm-hmm.
Are we going too far by aspiring for a future where malaria control does not involve pesticides? Are we dreaming too heavy?
No, I think you're not dreaming. In the first place, people who have a dream should always try to aspire and achieve that dream or realize it rather than giving up. I think that in 15 to 20 years' time, we will see a situation where pesticides really play a role in the background, from at least in malaria control, but possibly also in Aedes aegypti control for dengue and so on. And the reason that I'm saying is that alternative technologies are being rolled out now so quickly, with proof left and right, that it may well be possible that they will be overtaking the principal role of bed nets. The WHO is right. At the moment, there still is not a credible alternative that could protect so many people from malaria as bed nets do. But they're also, and look at the global vector control response, they realize they must go to integrated vector management in order to make it sustainable. And pesticides may have a role there, and it's hard to say whether it will be large or small, but it will definitely be drifting to the background.
There was, I believe 2 weeks ago, a little red publication coming from a Swiss group that looked at cognitive abilities of children exposed to agricultural pesticides. And what they meant by exposure was, have you smelled anybody spraying chemicals? Or have you been in an environment where they do this? And there were clear increases on things such as headaches, but also on cognitive capabilities. There were decreases, there were deficiencies associated with pesticides. So I think that even though people are not speaking about this right now, they will start to speak about those. But anyway, Willem, another thing that I pulled out, this is from your publication with Ann Wilson of the year, is this narration of the history of vector control. I have abrogated myself the role to classify into two, and on the left, if you look at it, clearly you see in the early days a lot of use of environmental management, and then all of a sudden at some point we go into commodities. And the question here I have for you is, where did this all go wrong?
Well, this went wrong in the early days, let's say from 1900 to 1950. There were no pesticides that you could use for vector control, at least not safely. And environmental management had been proven very successfully, particularly in Asia, to control vector-borne diseases, including malaria. So there was a big role until 1950, but then all of a sudden the miracle pesticide DDT appeared on the market, and it was also rather cheap. And we must admit that environmental management is time consuming and also needs to be repeated quite often. Sometimes you need to conduct drainage works, and so on. So when DDT appeared, for about 15 years, nobody wanted to think of anything else anymore. They didn't look back, and they said, this is the solution we must use.
Right.
Until they discovered it in Sri Lanka. They had a disaster in the coming because of the insecticide resistance and huge numbers of new malaria cases. That in many areas in South America, they had reinvasions of mosquitoes, and they needed to change the pesticides far more regularly. And this lasted till about the early 2000s, when all of a sudden money from the Bill Gates Foundation came in. They said, we need to do this differently. We need to develop entirely new pesticides with a completely new mode of action. And we're going to focus on 3 or 4 pesticides at the same time. And this program, which is actually headed by the IPCC, is likely to produce 2 or 3 new compounds in 2 years' time. But in my view, they're still making the same, and that's a commodity then. The industry can earn money with it rather easily, and the industry cannot earn money from environmental measures or building better houses. That money goes mostly to local builders, but not to very big industry.
Right.
And very big industries are very interested in continuing rolling out their products. So we have a bit of a struggle there to convince the big pharmaceutical companies and chemical companies to switch their thinking and say we need to go to integrated vector management.
Right.
And then it will not be only a method of commodities, but then I would rather say integrated vector control and cross out the word commodity.
Thank you. Thank you. Back to Anopheles funestus. I have pictures here shared by my colleagues from the Kilombero. All the 3 are the most common types of aquatic habitats for Anopheles funestus. At the bottom of the valley, you'll find the large ponds or the small spring-fed pools. On the escarpments, as you go south or north of the valley, you find the riverbeds. And this is a common phenomenon. You see this also in Western Kenya. Funestus breeding habitats. They're rare, yes, but you can find them. And so, I mean, in Tanzania, the government is very actively pushing for larval source management. And we're starting to think, how can you do this in rural areas? And it would be nice to hear your opinion on whether you think larval source management, either the environmental management way or just larviciding, would be applicable for these settlements.
Well, thank you for that question. I think larviciding has a big future. Why? Because if you have an effective larvicide like Bacillus thuringiensis israelensis at the moment, it will actually kill the mosquito in the cradle before it has a chance to build up adult populations. Right. Not many people realize that, but you immediately contribute to a lowering of the future mosquito population. Secondly, larviciding is something that anybody can learn. And farmers in particularly are very avid learners and very suitable to do that. We've just completed a study in Rwanda where we clearly show with Emmanuel Hakizimana and his group, where we definitely showed that you actually could reduce mosquito populations up to 90% in the rice fields in the highlands of Rwanda with BTI. And that was done by the farmers themselves. And we also completed a study in southern Malawi, where we had a community participation project, and the community themselves ran the larval control. What did we do there? the community participated in a couple of workshops, and then they created community action teams. And these community action teams would map from week to week the availability and the presence of breeding sites, and then decide this one needs treated this week and that one not. And so it went throughout the season. That had a fantastic effect on the mosquito populations. We were not able to measure that whether it had an epidemiological impact, but it's already enough if you can actually demonstrate that your mosquito populations go down. So I think there is definitely an additional role of larval source management.
Well, while we are, thank you for that, Willem. While we are on the subject of larviciding, it's also been mentioned multiple times that if you do larval source management very well or larviciding, you then don't have the challenge of outdoor biting or whatever, or insecticide resistance, because the mosquitoes are killed at source. On that point, our next question, this is the request of some of my colleagues, another tool that has been promoted a lot for control of mosquitoes that bite people outside as partial repellents. Now, without specifying any specific candidate, what are your general thoughts on partial repellents?
Well, I think the whole debate about spatial repellents and the proposal of using spatial repellents is a very good one, because it sort of can create a mosquito-free area immediately around the house. I don't think it works over a very large distance. The challenge here will be to find a candidate repellent that is so strong that it really keeps mosquitoes away from where you don't want them. And I think it's too early days to say at the moment how far the technology has gone and whether it has a very successful future or not. But I think spatial repellents together with an attractants, and then you get the push-pull combination, definitely is something that needs to be looked into very well. Unfortunately, the spatial repellents that are at the moment most looked at do not give sufficient protection, although they all show that they do chase mosquitoes away. But the only epidemiological study I'm aware of was just completed in Indonesia, in Sumba, and that was actually rather disappointing from the point of view of how much protection it afforded. So it's a technology that is in its infancy, should be pursued, and probably needs also quite a bit of financial input to discover novel repellents that we don't know of yet.
Just for the mention, one of the things we've tried in Bukhara, together with you, of course, is the push-pull, and in this case, seems that the candidates we have, that it is just better to have the spatial repellent on its own instead of having push-pull.
Yeah, that's what I believe. Yeah.
And what about? Go ahead.
The reason that you see that here, the repellent should be able to clear a couple of houses from mosquitoes, but at the same time you would like to get rid of these mosquitoes. So the pull system then has to function to actually act as a mosquito sink, removing them from the environment, so that the repellent actually can do a better job. Okay.
What about ATSBs? What are your thoughts? Attractive toxic sugar baits. This is also perhaps the most talked about new tool right now.
Yeah, it's to tell you frankly, Fredros, it's a very good tool for the areas where it was developed for. And that was Israel, and the edges of the Sahara Desert in the Sahel,
Mm-hmm.
in Mali, Burkina Faso, and Senegal. Because you have long periods of the year when it's extremely dry, and the mosquitoes actually then feed on plants. And if you have an ATSB, you can actually direct them there. But I still question how well it would work in an optimal ecosystem like in central Tanzania or Kenya, where the ATSB should be highly competitive with natural floral sources. And I still have not seen the studies where that kind of competition is clearly actually shown.
Yeah.
So it sounds like a good idea,
Yeah.
but I'm a little bit surprised about the excitement that it brings along.
Yeah, there's quite a lot of talk about it. My colleagues, Eric is on the call, and I know he's very heavily involved in this kind of trials.
Yeah.
But it's surprising how well it's worked in Mali. I mean, the graph at the
absolutely,
bottom
but once again, the mosquito environment in Mali,
right? Yeah.
Cannot be compared with the mosquito environment in Tanzania or Kenya. In Kenya, it might perhaps work around Baringo or a bit further north, where you have these very dry areas.
Yes.
So it should definitely be tested there, because then you may be very successful, and it's rather cheap.
Yeah. I want to ask you another question, another new tool. Zoo prophylaxis, zoo potentiation, use of insecticides given to animals. This is a picture from my mother's house many years ago. What do you think about this?
Well,
worth trying?
No, I think it's worth trying in areas where livestock is very much an important part of the economic system.
Mm-hmm.
But again, you expose the mosquitoes to insecticides like ivermectin. It is another chemical, and the question is simply, how long does it last before you have selection against these chemicals, and then you're again standing empty-handed, because the mosquitoes can feed on the cattle. If they don't die, they all will produce offspring. So that doesn't help you very much. So as long as it works, yes, do not put all your eggs in this basket, in my view.
That's an interesting view. I mean, we have some of my colleagues here working also on insecticides. And I mean, a common question has always been, is resistance going to develop against this, especially metabolic resistance? So it will be important to see what the next few years bring in that regard.
Yeah, if you go back to the whole history of zooprophylaxis, because this is not new, of course, it was actually one of the first alternatives of malaria protection that was already suggested way back in the 1920s. It has never actually been extremely successful to the extent that people said, yes, we need to do it. Now, since we have seen these studies with ivermectin, primarily in West Africa again, it was definitely shown that mosquitoes that had fed on animals that have ivermectin are dying, yes, or they don't produce offspring anymore. But again, in my view, it's not much different of exposing mosquitoes to a bed net than to an animal with ivermectin.
All right. Actually, we spoke to one scientist who told us that there might be value in retaining Anopheles arabiensis, which is the most dominant animal biter, because you can then use them as a biological control for an office gum because they occupy that niche, which is a very interesting hypothesis. But yeah, it's nice to hear that. Now, Willem, I want us to shift a little bit from the technical versions of our discussion. Go ahead.
Just one more question.
Yes.
My time is almost running out.
All right. Oh, okay.
So let's see that we can at least handle the most important questions that are still hanging in there.
Yes. Okay, I get that. Let's just do that. This one is important, and then we'll skip there.
Yeah.
Should we worry about this, or can we just balance genomics and field entomology?
We should be able to balance that,
right?
Because if we don't do, in my view, we fail the countries where these endemic diseases exist. But what we absolutely must make sure is that the education systems continue to teach biology, entomology, parasitology, and not only praise that biotechnology or molecular studies are the future.
Thank you, Willem. As we draw towards the close, I want you to talk about two things as we close. One of them is public engagement, and the other is capacity building. And during your conversation, you have mentioned a lot how important it was to engage with the communities. And it would be nice, just based on your own experiences working in different places, what some of the strengths and challenges you have encountered in the process of trying to engage governments or local communities in disease control, vector control?
Okay, let's start with government, because in many countries, the control of vector-borne diseases is still led from the Ministry of Health, in a rather vertical way. Decisions of how to do it are being decided upon at the Ministry of Health, and they, of course, ask for advice from people in the field, but very often they arrange the money, take the core decisions, and that's not only in terms of drug distribution, provision of medical staff, but also for finances for vector control. But governments have for far too long actually completely ignored the will of the people, of the stakeholders actually. And in my view, you just can't enter a village or a community and say, well, we know you have malaria. We're all going to give you bed nets next month. Be ready for it. In my view, you would have to almost do it turn the whole system around. And first explain and make sure that the population is totally aware of their health situation, what causes it, and what they can do, for instance, themselves. And I think what we talked about earlier, the house improvement program and schedules must be an eye-opener to communities. Why didn't we know this earlier? That if we make our houses mosquito-proof, that already most of the problem could be gone. And secondly, I've been in several countries where bed nets were supplied by the government and they were never used because they were of the wrong color or the wrong shape, because nobody was ever asked what they actually wanted. And fortunately, this is changing rapidly. Also at government level, all of a sudden people are discovering, hey, community participation and community involvement is almost critical for the success of our program. To be carried actually to where it should be. So I think, what are the strengths? It's clear. If people, and you see a beautiful example here, somebody explaining to a lady probably what the procedure is going to be for an intervention, that already helps tremendously. And then people say, nobody has ever done that for us. We always were asked if we had questions, we had to come to the clinic, and then they would tell us, oh, you don't feel well, here, get a few tablets.
Right.
Or we hand the bed nets out and you sort it out yourself. And that is rapidly changing, fortunately, to the advantage of communities and government.
Thank you. So, I mean, for the sake of time, I'm going to move this to the last question, which is really about, for the benefit of my colleagues and myself, we want some advice, Willem, on young people. What would you tell young people just getting into medical entomology today?
In the first place, if you want to get into medical entomology, you're interested into medical entomology, try to find out which insect you like actually best. You don't need to stick to mosquitoes. There are flies, there are ticks, there are bugs, all kinds of things you can look at. Mm-hmm. And also realize before you continue whether you like being out in the field or not. Some people clearly admit, no, I'm not actually fit for getting dirty or working long nights. I prefer actually a clean lab environment.
Yeah.
So just be aware of that. But other than that, once you are interested in a particular topic, just pursue that one and don't give up. It's only those people who are really convinced that they say, for example, oh, I'm so interested in the interaction between a mosquito and a parasite. How can I get the best knowledge? How can I do experiments to unravel that particular situation? And they usually get where they want to, because there are enough people like you and myself who can give suggestions where you can go for training, what opportunities there are to get a grant. And not everybody needs to get a PhD. That's often the mistake. Often already with a Master of Science, you can get a very nice position and a very important position wherever you want to be. And in my lifetime, and I'm really happy to see that, we've seen this major transition that all over Africa, young African entomologists actually became so well trained and proud of themselves that we of the old guard have almost been phased out, fortunately. And you can actually be very proud of your own nation and continue with the knowledge you have.
Yeah.
But in order to make it work, you must collect the data yourself or know how the data were collected.
Thank you. Thank you, Willem. But for you also, I mean, and that's why I want to end by saying, you've just been talking about, you know, how nice it is to see these young African people and scientists develop into reliant scientists. But I believe I have, as one of the beneficiaries of the system, I mean, one of the main reasons that I got into this business is yourself. And I just want to recognize the value that Wageningen University and Research Center has played in the development of medical entomology in many, many parts of Africa: Nigeria, Mozambique, Kenya, Uganda, Malawi, and several African countries. In terms of capacity building, many years ago, I met a friend of mine, Rafael Ngesan from Benin.
Yes.
Who was a technician until he grew old. And then he said it is because he went to Wageningen that he was able to put his papers together and get a PhD and go back home and get a respectable position. And you've done this with many, many people. I am very, very grateful for you, Willem. Thank you so much for yourself and your team at Wageningen. for all the contributions you've made to capacity building in Africa and to the rest of us. Thank you so much.
Well, thank you, Fredis, very much. I've enjoyed it thoroughly. And I wish you all actually a very successful future. I can't see most of you, but I see how many actually participated this afternoon. So it was very nice to know. Thanks very much, Fredis, for organizing this, and we'll talk again.
Thank you so much, and I will share with you an audio recording of this as well. Pass my regards to family and to my colleagues on the call. I'm not at the same location as you, but I hope you did enjoy this conversation as much as I did. And on behalf of everybody at Ifukara, thanks to Willem, and we bring this to a close. Bye-bye.
Okay, thanks very much.